In today's session, I am going to talk about animal classification. Before we actually begin with the classification and the details of it, let's first of all try to understand as to why do we need to classify. Whether animal, plant, microorganism, whatever, why do we need to classify? The first point under that is it gives you a systematic approach towards understanding, towards studying, towards researching on a particular living organism, a plant, animal, whatever. Now, what do I mean by systematic? Systematic means you start, the process of classification starts from a larger group to a smaller group. So if you know what the smaller group you are going to study, you can get an idea as to what it has come through. That group has originated from which particular larger group, all that you come to understand, and all those characteristics you get to understand from it. So it gives you a systematic approach. Secondly, it gives you a phylogenetic relationship. Now here I may mention that when you start classifying a particular group of animals, group of plants, whatever, you have a specific way of placing them. Which type of plants first, which type of animals first, which type of animals second, third. That is specific way of putting them, and that is based on their phylogenetic relationships. Now, what is phylogenetic relationships? The lesser evolved to higher evolved or better evolved. That is how the system goes, and the relationship is called as phylogenetic relationship. So by classification you establish a phylogenetic relationship amongst them. Thirdly, each taxon. So each taxon in classification carries a few specific descriptions, a few specific characters. So by understanding taxon of the organism that you are trying to study, you get to know about a few characters that go along. Say for example, if I am studying a particular vertebrate, then that term vertebrate itself carries a few characters. Like it has to have a vertebral column, it has to have a cranium, it has to have red blood corpuscles, it has to have bilateral symmetry. So many features, they go along a single term, that is vertebrate. So with the taxon you get to understand these. Next is, by classification you assign a naming for a particular living organism which is universally acceptable. What do I mean by universally acceptable? If I am talking about, just as I mentioned, vertebrate, then that term vertebrate would have same single meaning world over. Whether you write it in vernacular language, you write it in Japanese language or whatever, that name vertebrate would have the very same meaning. So classification gives you naming an organism which is universally the same, universally similar, universally acceptable. Whatever organisms we know about, if we classify them, if we name them, then it becomes relatively easier for us to identify if there is a new species, if there's a new organism that has come up, which has not been described, which has not been identified. This becomes easier because all earlier ones have already been classified. That's one more advantage. Now in classification you give, as I said, a name for every organism, and there is a double name for every organism. That is called as a binomial nomenclature, a system that was introduced by Carolus Linnaeus. He was the one who introduced the system of binomial nomenclature. Now, what is this system? The system says that the first name of an organism, it stands for its genus, and the second name stands for its species. But that's not all. Binomial, bi means two, norms means rules, regulations. There are two rules to be followed. And what are the first rule says that genus name has to begin with a capital letter, species name has to begin with a small letter. And both the letters or both the names have to be either italics, that is, if you type that, they should be in italics, or if they are handwritten, they should be separately underlined. These two rules have to be followed for naming every organism. So binomial nomenclature gives you a universally acceptable method of naming rather. The interesting part is his name actually was Carl von Linné. But when he introduced this system, he changed his own name into Carolus Linnaeus. Homo sapiens, Mangifera indica, Apis domestica, that kind of naming system, he introduced his own name as Carolus Linnaeus. That's the interesting part of it. Now with this method, naming method and further classification, you assign a particular position to each living organism in the entire living world. Just like we assign a particular location with GPS, latitude, longitude. Same way you have two names here, one comparable to latitude, other comparable to longitude, and that specifies a particular position for a particular organism. So you define a method of positioning each living organism in the entire living world. These are the advantages of classification. Now let us get to classification of animals. Before we actually start with the classification process, we need to get acquainted with a few related terminologies. First of all, I will talk about germ layers, or they are also called as blastoderms. Blastoderms because derma means skin layer. Blast stands for fission, division of cells. So layers formed by division or multiplication of cells, hence they are named as blastoderms. That means, see, every multicellular organism starts developing from one single cell, either a zygotic cell or whatever. That cell undergoes repeated mitotic divisions to give rise to the organism under consideration. In that process, there is a stage reached when there is a large cavity covered over by a layer of cells. That layer of cells multiplies further into 2 layers, 3 layers, whatever. These are the blastoderms. These are the germ layers. Now, why do we need to know about them? Suppose if this is some animal, this is its alimentary tract, digestive system, digestive tract. Now, if I take its section, you can call it vertical section or transverse section. Then the possibility is, first of all, I would see the outer layer as one single circle. Alimentary tract as another circle. If I see only these two circles, then the outer circle will be named as ectoderm, inner one as endoderm. Endo means inner, derma as I said means skin. So inner covering, outer covering. If only two layers are there, like what you're seeing in this figure, then that kind of organism is called as diploblastic. Diplo means two. Blastic stands for blastoderms. Two blastoderms, hence the name diploblastic. Another possibility is this is the outer layer, this is the inner layer, and one more layer is possible here. That additional layer which you are seeing is called as mesoderm. Meso means middle. This is another possibility. Now you're seeing three layers, so that's called as triploblastic. Triplo means three blastoderms, hence triploblastic. But that triploblastic also has two possibilities. One is this is outer layer, ectoderm. This is inner layer, endoderm. And this is mesoderm. Now this mesoderm and this mesoderm, there's a slight difference. Here is a double mesoderm, single mesoderm. There's another related feature that we need to consider. This cavity here, bound externally by ectoderm and internally by endoderm, is what is called as a body cavity. Cavity, large cavity inside the body, hence name body cavity. In the first figure, if you observe, the body cavity is covered externally by ectoderm, internally by endoderm. The second figure, it is covered externally by mesoderm and internally by endoderm. While in the third figure, it is covered on all the sides exclusively by mesoderm. Externally, internally, it is in contact only with mesoderm. This kind of body cavity, it's called as coelom. Coel means a cavity. It's a true body cavity. It's a coelomic body cavity, which represents a body cavity that's covered by mesoderm on all the sides. Externally, internally, it is exclusively in contact with mesoderm. That kind of body cavity is called as coelomic cavity. One thing that goes wrong, this is possible only in those animals which are triploblastic. In diploblastic animals, there is no mesoderm, so coelom is not possible. So triploblastic animals can have coelom. Mind well, this is also triploblastic, but that's not coelom. So triploblastic can be with coelom or without coelom, but diploblastic can never be with coelom. Another feature, another related phenomenon that we need to understand is symmetry. If there's one single plane of symmetry, consider a frog, consider a fish, consider human being. There's only one single plane of symmetry that can divide the animal into two exactly symmetrical halves. Then you can call it as bilateral symmetry. Bi means two, latera means sides. Only two symmetrical sides are there, hence it is bilateral symmetry. Then if you consider a starfish, along the radius you can make more than two symmetrical parts. We call that as radial symmetry. Then an animal like a snail, if you consider, with that coiled shell, it cannot be divided into symmetrical parts, neither in one plane nor in more than one planes. Such animals are called as asymmetrical animals. There is no plane of symmetry. There is one more type of symmetry, that's called as spherical symmetry. Imagine a ball, cut it vertically, two equal halves, horizontally two equal halves, obliquely two equal halves. That's called as spherical symmetry. But keep in mind, spherical symmetry is not exhibited by any animal as such. It can be seen in the larval stage or it can be seen in a colony, but an individual animal would never exhibit spherical symmetry. These are the types of symmetry which we need to keep in mind. Let's then go to talk about the actual classification of animals. First of all, let's talk about biosphere. Now, how do we understand the concept of biosphere? See, if I talk about hydrosphere, it represents that part of the globe or the earth which is occupied by water. Atmosphere, that part of earth occupied by air. Similarly, biosphere stands for that part of earth or this globe which is occupied by bio means life, living organisms. So part of sphere, part of globe, part of earth occupied by life is biosphere. So that will include all kinds of organisms, all kinds of living organisms, which we would first of all separate into unicellular and multicellular. One single cell organization and made up of more than one cells. That's how we distinguish unicellular and multicellular. Unicellular would further be divisible into prokaryotic, that is primitive nucleus, undefined nucleus, uncovered nucleus. They would come under monera. Now what do we call this as? We call this as kingdom. Largest group in this classification process. Monera would include prokaryotic unicellular organisms. Second one would be protista. That would include eukaryotic unicellular organisms. Eukaryotic means the nucleus is well defined. The cells are better developed. And multicellular would be further divisible into three kingdoms: plantae, fungi, animalia. How do we distinguish? Multicellular autotrophic, multicellular saprophytic, and multicellular heterotrophic. That's how we distinguish three different kingdoms under multicellular group. I am going to focus this session mainly on a bit of protista and entire animalia. Okay. Monera, plantae and fungi would not be considered in this section. Okay, under protista there is one phylum which we will consider, and that is Phylum Protozoa. And then, as I said, we will be talking about kingdom Animalia. This kingdom we would divide into two subkingdoms. Parazoa and Metazoa. See, Protozoa would be meaning proto is primitive, zoon is animals. So primitive animal-like unicellular organisms. Why animal-like, that we will consider in the later course of time. Animalia is separated into two subkingdoms, Parazoa and Metazoa. Parazoa is a kind of multicellular organism, but these cells are not centrally coordinated. It's just a kind of aggregation of cells, collection of cells, gathering of cells. Different kinds of cells, different structures, different functions, but one single organism is formed with them. That's Parazoa. While under Metazoa, at least tissue formation would be there. That means structurally and functionally similar cells coming together to form a tissue. That is the minimum level of organization which will be seen in Metazoa. Under Parazoa, again there is one single phylum. Phylum Porifera. Metazoa would be further separated into diploblastic animals and triploblastic animals. Under diploblastic, again there is one single phylum. Under triploblastic, we would further make three divisions. A coelometra, that is triploblastic but without coelo. Pseudocoelometra, that is triploblastic with false coelo, and coelometra, that is triploblastic coelomate animals. That's the kind of classification we are going to follow. Now, under Acoelometra, there's again one phylum we will consider, that is Phylum Platyhelminthes. Platy means flat, helminth means worm, so Platyhelminthes. Under Pseudocoelometra, again there is one single phylum. I'll write the name here. Phylum Aschelminthes or Nemathelminthes. Aschel means a cylinder, so these are cylindrical worms or roundworms they are called. Under Coelometra, there will be a number of phyla. We will talk about them in due course of time. So that's a broad outline of division of Protista and Animalia. That is Kingdom Protista and Kingdom Animalia. Details of each phylum we will take up in further course of time. So as I said, this is the whole chart showing classification of animal kingdom. Now in the previous part, as I said, this is a 5 kingdom system which we are following, and this was introduced by Robert Whittaker, whereas the 2 kingdom system, wherein Monera, Plantae, fungi would be together. That will be identified as kingdom Plantae. And Protista, Animalia together, that will be identified as kingdom Animalia. The two kingdom system was introduced by Aristotle, the great philosopher, somewhere in 384 to 320 BC before Christ. While the five kingdom system was introduced by Robert Whittaker somewhere in 1969, quite a recent one. So in the current sessions on animal classification, I will be following the five kingdom system, which is the relatively recent one. As a seed outline of classification we have seen, under unicellular group, unicellular category, there's one kingdom Protista, under which there's only one phylum Protozoa, which we would take into consideration. Protozoa. Proto means primitive, zoon means animal. Let's take this as primitive animal, but we have to understand that these are not animals. This is a separate kingdom that includes only unicellular organisms. While animals, we have identified them as multicellular heterotrophic. So these could be heterotrophic, but these are essentially not animals. Still, we include them under zoon term. A very simple reason for that is these are animal-like cells. What I mean by that, when you distinguish between plant cells and animal cells, the prime difference is presence or absence of cell wall, presence or absence of plastids, presence or absence of lysosomes and all that. So these cells are more like animal cells, hence zoon, primitive animal-like unicellular organisms. Now the term unicellular at times can be replaced with acellular, because unicellular, if I have to say, I would have to say it's made up of one cell, but it's not made up of one cell, it's like one single cell. So a better term would be acellular, wherein the cell and the organism are not separable, it's one and the same thing. So these can also be called as either unicellular, that's a convention, or acellular. So whatever functions the organism has to perform, they would be restricted to that single cell. Each cell is an independent organism. Now what are these functions? First of all, I see these are animal-like cells. So cell wall would certainly be absent, made up of cellulose, hemicellulose, all that. But still, the cell membrane may further be covered with something called as pellicle. Maybe all the protozoans are not with pellicle. Some are with pellicle, some are without pellicle. Pellicle gives it more or less a fixed shape, which may be seen in some of the protozoans, or some of the protozoans may not have a fixed shape. That will depend basically upon presence or absence of pellicle. Usually these are single cell. Single cell, what I mean, solitary. Solitary, one individual, second individual, they would be staying separately. But rarely they can show colonization. Number of organisms come together, stay together. Mind well, it is not comparable to parazoa. They stay together as a colony. Each organism is an independent organism. Each cell is an independent organism. Next to pellicle, inside the pellicle rather, is the cell membrane, and inside the cell membrane, whatever plasma, whatever cytoplasm is there, it is clearly distinguishable into ecto and endoplasm. Ectoplasm and endoplasm. That is, ectoplasm is non-granular but more fluid kind. Ectoplasm is relatively thicker, denser, jelly-like and granular. These are, in most of the protozoans, these are clearly distinguishable. Ectoplasm is distinct from endoplasm. Mode of nutrition, if we consider, these could be holophytic or holozoic. The word holo stands for entire. The organism would be engulfing the entire. Phytos stands for plant and zoon stands for animal. Now here what I mean by plant is a plant-like microorganism. Could be a bacterial cell. So holophytic or holozoic, animal-like organism, smaller organism. Entire organism would be engulfed and would be digested inside the cell. That digestion inside the cell, it is by formation of a food vacuole, which will undergo a regular process of digestion. Digested substances will be absorbed in the cells, undigested matters will be egested from the cell. Okay, that is how the nutrition would happen. Now that movement of food vacuole from the place of engulfing the organism till the place of egesting the undigested matters, that may follow either a definite route or an undefined route. It will depend upon the specific organism that's under consideration. Okay, so it's the food vacuole which will help in ingestion, digestion and elimination of undigested matters. That's the mode of nutrition, you can say. When you talk about vacuole, there is something called as a contractile vacuole as well. As the name suggests, it can contract and relax. It can contract and extend. This would help in maintaining an osmotic balance. So let's call it as osmoregulation. So it's a vacuole which can contract and relax so as to maintain an osmotic balance. Salt balance, you can say, water balance, you can say. Concentration of cytoplasm with respect to the surrounding medium has to be appropriately balanced so as to avoid excessive loss of water or excessive gain of water. That is by a phenomenon called osmosis. That is taken care by the contractile vacuole. Okay. Now, structure and functioning or mechanism of the contractile vacuole may vary depending upon the organism under consideration. But broadly, it's a vacuole which can contract and relax so as to maintain an osmotic balance. That is how we can describe. Locomotion. See, it's a single cell, but still is capable of undergoing locomotion, which may be with the help of pseudopodia. Pseudo means false and podos means appendage. False appendages, false extensions comparable to appendages would be there that would help it in locomotion or at times even nutrition. We would talk about that in due course of time. So locomotion could be with the help of pseudopodia or could be with the help of tiny hair-like extensions called cilia or relatively longer extensions called flagella. So locomotion either with the help of pseudopodia or with the help of cilia or with the help of flagella. Some of the protozoans may be non-motile as well. We will talk about that in due course of time. Reproduction. Reproduction can be by binary fission. Binary fission. Fission means cleaving, division. Binary means one single cell, one parental cell dividing, mitotic division, mind well, dividing mitotically into two daughter cells. But here the cell itself is an organism, so one organism, one unicellular organism divides into two unicellular organisms. Mitotic division, so chromosome number would remain the same in parent and daughter cells. That's binary fission. Or it could be by multiple fission. That is, one parent cell divides into more than two daughter cells. A large number of daughter cells, daughter organisms will be formed, which can then be released and can develop into adults. That is by multiple fission. Or it could be by conjugation. Conjugation would mean two cells will come together. A cytoplasmic bridge would be established between the two. Nuclear contents will be exchanged. Nuclear contents will be exchanged, not the whole. Part of the nuclear contents from one cell would be exchanged with part of the nuclear contents of another cell. Then the cells would separate and would start undergoing again binary or multiple fission, whatever. So in conjugation, what happens, rather than reproduction, it's a method of exchange of nuclear material, exchange of DNA, exchange of chromatin. That can take place in some of the protozoans. And some of them even show a process called encystment. Now let me tell you, encystment would not be a mode of reproduction. Encystment would be a mode of protection of the cell. A highly protective covering develops around the cell, around the organism, inside that covering, inside that cyst, the cell, the organism will undergo either binary fission or multiple fission. That cyst would be a dormant stage, a resistant stage. So once again, when that cyst comes across a suitable condition, a favorable surrounding, that cyst will disintegrate and the tiny organisms, the tiny cells will then start developing further to become the adult cells. Process of reproduction may continue. This is a broad outline of what protozoa is. It is divided into 4 different classes. They are Rhizopoda, Celiophora, Mastigophura and Sporozoa. All of them would be unicellular, that's applicable to all of them. Most of them are freshwater dwellers with 1 or 2 exceptions. Most of them are solitary, singular. Rhizopoda, that term comes from rhizos means root and podos means appendages. That means root-like appendages. What does that mean? They would be showing pseudopodia for locomotion. And these pseudopodia, in addition to functioning as locomotory extensions, they would also help in ingestion of certain solids along with liquids, or certain liquids as per the requirement of the cell. So intake of substances along with water, which is comparable to absorption by roots, rhizos, hence the name Rhizopoda. Pseudopodia, when I say, it has to extend its body to form the structures, so pellicle would be absent. The shape of the cell is not fixed, not defined, variable shape. An example under this can be considered as amoeba. Irregular shaped, variable shape, changeable shape. Pseudopodia would be formed as locomotory organs. One single nucleus right in the center. Ectoplasm, endoplasm is clearly distinguishable. Food vacuoles, contractile vacuole also would be there. These are freshwater dwellers. Reproduction is majorly by binary fission. Exceptionally, even multiple fission would be seen, encystment also is possible, but we don't see conjugation here. That's Rhizophora. Ciliophora, as the name itself suggests, cilia, phora means to bear. Bear cilia as a locomotive organs. Okay. The shape is fixed. That obviously means covering of pellicle would be there. I can talk with an example. Paramecium. Cilia as the locomotive organs. And if we're talking specifically about paramecium, which has a slipper-like shape, cilia all around the body. Here the cilia would be relatively longer. Now this is a protozoan with two distinct nuclei. A more or less kidney shaped meganucleus, mega means large, and small spherical micronucleus. Two prominent contractile vacuoles will be there. Here is mouth of the cell. Now this is broadly what paramecium looks like. The cilia all around the body, they would be there. They would help in locomotion. Mind well, the direction of locomotion, its anterior portion is here, the blunt one. So it moves in this direction. When it's moving, you can see a small opening like thing. That's called as a cytostome. Stoma means mouth, cytome means cell. Mouth of the cell through which the food organisms, the food particles rather, can be ingested, and they would give rise to formation of a food vacuole here, which can then move through the cytoplasm, contents will be digested. That is how the nutrition takes place. Contractile vacuole helps in regulating the water balance, maintaining the water balance. How is done? There is a central vacuole extending from which would be the radial canals. This is what it looks like. If it has to absorb water from the cytoplasm, that water will be absorbed through these. The central vacuole will grow in size. And if it has to release water into the cytoplasm, the central vacuole will reduce in size and the water will be given out through these radiating extensions. That is how it would function for maintaining the water balance. That's a contractile vacuole. Now, as I said, there are two nuclei. Meganucleus eats a vegetative nucleus, and micronucleus is reproductive nucleus. All vegetative functions, nutrition, locomotion, all those will be under the control of vegetative nucleus, that is meganucleus. Reproductive activity will be controlled by reproductive nucleus, the micronucleus. And this animal does show the process of conjugation. That is, two paramecia can come together, a cytoplasmic bridge can be established. We can have it here. This is one paramecium. This is another paramecium. A cytoplasmic bridge can be established. The micronucleus now has a role. It will divide into two. This would come here, this would come here, then they would separate and would start undergoing regular binary fission. And here binary fission is transverse binary fission. Transverse binary fission. And just before binary fission, all these structures the cell has got, they would be doubled. Contractile vacuoles will become 4, cytostomes, cytopharynx would become 2, and then the cell would divide. So that resulting daughter cells will have the same structures as the parent parent cell, parent organism. That is how it usually multiplies. But after a specific number of multiplications, binary fissions, it goes for conjugation. This kind of conjugation. So that its hybrid vigor can be restored. Its possibility of survival is increased. That is what you see in Paramecia. It's also a freshwater dweller. The next class is Mastigophura. The word mastigos stands for a whip. A whip-like flagellum is provided for locomotion. So here the locomotion is by flagellum. Example can be taken as Euglena. A structure is something like this. This is the flagellum. Here is the nucleus. It's a tiny spot, and this invagination is called as a reservoir. This is named as a reservoir. Uniquely, Euglena is provided with plastid. Chloroplast it has got. So it can synthesize carbohydrates to some extent. But that doesn't satisfy its nutritional requirement. So through here, the food particles along with some water can be ingested. It can result to formation of a food vacuole, which can then undergo digestion inside the cell. So nutrition is autotrophic as well as heterotrophic. Okay. Rather, this is called as a phytomastigophoran. Phytomastigophoran, because it has a plant-like feature, presence of plastids, presence of chloroplast. So phytomastigophoran. There are other mastigophorans which are zoomastigophorans, in which plastids are absent, chloroplast is absent. Flagellum helps in locomotion. Now here reproduction is predominantly by binary fission, and it is by longitudinal binary fission, unlike what we saw in Paramecia. Here it is transverse binary fission, here it is longitudinal binary fission. To begin with, the flagellum will divide, then the nucleus will divide, then the cell will divide, so that 2 euglenae will be formed. That is how the multiplication would happen. Then lastly we come to Sporozoa. Now here there is no mention about cilia, flagella, rhizopoda, pseudopodia, whatever. That means here locomotory structures are absent. These are majorly parasitic organisms. And being parasitic, they do not need any specific locomotive organs. There is usually a vector which will carry the organism from one host to another host. Hence, locomotive organs are not found to be present here. Example could be Plasmodium, plasmodial parasite, you can say, a malarial parasite. Different stages are there. A sporozoite stage could be something like this. That is an invading stage which enters the human blood and then undergoes a vegetative phase of its life, asexual phase of its life inside the human body. Then it enters mosquito, where it completes the reproductive phase. So here it doesn't need any specific locomotory structures because it's going to get carried through a vector. Obviously, all its structures are as per its body physiology. So no contractile vacuole, no locomotory structures, no specific protective pellicle would be there because it has all adaptations for being resistant in the environment where it survives. Okay, So Protozoa would be the fourth class under phylum Protozoa. I'm sure you must be enjoying these videos, so do put a like, subscribe to the channel, share these videos among your friends and family. And if you have any suggestions or recommendations or queries, do not forget to post them. Thank you.